As you prepare for the final, let's make sure you're clear on the correct answers for short test 1. In general, the class did pretty well, although the responses for questions 4 to 6 were a little bit weaker than those for the first 3 questions. So let's have a look at all the solutions to make sure you're on track for the final. Question 1 was simple definitions taken directly from your lecture notes. It's important that you understand the differences between these terms, as they have direct implications for being able to understand survey design and also how to determine which statistical test you should use in a given circumstance. Nominal data is often also referred to as categorical data. Categorical data are used simply as identifiers or names, so things like color, gender, or species. On the other hand, ordinal data is an ordered series of relationships or rank order. So individuals are ranked in some way. However, it's important to note that if ordinal data is numerical, the numbers are not a measure of quantity. A good example of this is placings in a race, where participants are ranked first, second, third, etc. However, the time between each of these placings is not necessarily the same. A response variable is also known as a dependent variable. This is typically the thing you are specifically investigating. An example could be growth rate of tomatoes that might be subject to different conditions. An explanatory variable is also known as the independent variable. These factors are measured and monitored as they are likely believed to have some impact or relationship with the response variable. The target population is the complete set of units in the population which you might take a sample from, and sometimes it's referred to as the sampling frame or perhaps the target audience. The experimental unit is the unit that receives a treatment in a lab setup. In a field setting, it is the unit that could be characterized by having a given level of factors or combination of factors. The experimental units must be independent of each other. Paired data is when repeat measurements have been made on the same experimental unit, and so they cannot be considered independent. This could be left and right hands, leaves from sun and shade areas on the same tree, or it could be before and after a pollution event. Non-paired data occur when you've got 2 independent experimental units present and data has been measured on both. So you could be measuring at different sites, on different species, etc. For question 2, you were given a list of squares that were sampled and then asked what type of sampling method was being used. So the best thing to do is to mark these squares so you can get a sense of which ones were chosen. When you do this, you'll see a regular pattern that will hopefully then lead you to decide that systematic sampling was done. An advantage of systematic sampling is that it might be easier to locate samples, especially for experimental units that aren't easy to mark individually. In general, systematic sampling is less intensive as well, because you know exactly where you're going to sample your cells and it occurs at a regular rate as well. A disadvantage of systematic sampling is that by chance you might be systematically sampling a confounding factor that is present at your field site. So for example, column 1 might be an area of sloping land. So if systematic sampling were used, you might have a disproportionate number of samples from sloping areas. Other types of sampling methods you could have described are random, cluster, haphazard, or stratified. Definitions of all of these are in your lecture notes. Question 3 was well done by pretty much everyone, so I won't talk about it here, but I have listed the solutions so that you can confirm all of your calculations. Question 4 is where some of you got into trouble. The first and very important thing is to read the question carefully so you know exactly which question you are being asked. When you do this, you'll see that you are specifically being asked to run a F-test. So, in order to do this question, you must know what an F-test is testing and how to calculate an F-value. So be sure that you realize that an F-test is about variance, and in particular, comparing the variance of 2 samples. The null hypothesis for this question is the variance of the length of snail shells collected in Rotuma is equal to the variance of the length of snail shells collected in Laos. The alternate hypothesis would simply be the opposite, where you'd insert not equal instead of equal. When estimating the degrees of freedom for an F-test, remember that two different numbers are required. One for the numerator and one for the denominator. The degrees of freedom for the numerator will be the sample size minus 1 for the sample with the largest variance. The degrees of freedom for the denominator will be the sample size minus 1 for the sample with the smaller variance. The data given to us in this question indicates that shell length of those individuals collected in Laos has larger variance, i.e. 2 millimeters squared, and so we use the Laos sample size of 25 to determine the degrees of freedom for the numerator is 24. The Rotuman sample size is 30, and so the degrees of freedom for the denominator is 29. To calculate the F value, simply divide the larger variance by the smaller variance to come up with 2.5. To make an interpretation, first check the F table to see what the critical value is for the given degrees of freedom. When you do this, you'll see that 2.15 is the critical value. So the interpretation would be the variance of the length of snail shells found in Rotuma is significantly smaller than the variance of the length of snail shells found in Laos. You have an F value of 2.5, degrees of freedom of 24, 29, and a p-value of less than 0.05. The type of test that would be then chosen would be an independent t-test with unequal variances. This is because the question told us that two samples were normally distributed, and our F-test just told us that the variances were not equal. Question 5. First thing to do is make sure you pick up all the information given in the question. So looking at it, you can see that you've got just one sample and that measurements were made on 30 individuals, and so n equals 30. The average value that was found was 2.45 kilometers squared, and there was a standard deviation of 1.25 that was associated with this value. The value that you are testing against is 3 kilometers squared. First thing you were asked to do was to state the null and alternative hypotheses, and these are listed here. Make sure you are as specific as possible. You must refer to average values, you must refer to male Samoan flying foxes in Viti Levu, and you must state what you are testing against, i.e. a value of 3 kilometers squared. Looking at the critical t-value, you will find that for 30 minus 1 equals 29 degrees of freedom, a p-value of 0.05 and a two-tailed test, that the value is 2.045. Running a one-sample t-test, you should get the result as shown. You will note that this value is greater than the critical value. Remember, a negative value should be compared to a negative critical value. Our interpretive sentence becomes: the average home range of male Samoan flying foxes is significantly smaller than 3 kilometers squared. The t equals negative 2.41, degrees of freedom equals 29, and the p-value is less than 0.05. Make sure that you mention what you're actually testing, and also give the t-value, the degrees of freedom, and the p-value. In question 6, you are given some R output to decipher. So before looking at the questions, let's take a look at what the R output tells us. You can see we've obviously got two sets of data. Normality was tested for each with a Shapiro-Wilks test. Since these were tested separately, we know that the data is not paired. For both data sets, for the Shapiro-Wilks test, the p-value was less than 0.05, so we can say that each of them was not normally distributed. Finally, a test statistic is shown. You were then asked what type of test was run. As just said, we know we've got two samples that are not paired and are not normal, so our only option is a Mann-Whitney U-test. Reasons you could have given for choosing this could have been any of the three that are listed there. So the fact that there's more than one sample, the data is not paired, individual variables are not normally distributed, and that the test statistic given was W. You were then asked whether you would accept or reject the null hypothesis. In this case, you would reject because the p-value is 0.01383, obviously less than 0.05. The types of summary statistics you would use for this problem would be ones that are appropriate for non-normal data. So that would be the median for central tendency and the range or the interquartile range for variability. For the final part of this question, you were asked to draw a frequency table of the control dataset. So what you needed to do was tally all of the values from this dataset into a table with given bins or ranges of values, along with the associated counts for these bins. Here I divided the dataset up into 50 values per bin. You could have chosen a different number of bins, however fewer than 5 and greater than 10 are not really good options. The other thing is that the size of the bins, i.e. the values covered by each, should be equal. So there you have it, how to ace short test 1.